Today's study tip, after an exam, try to go back and think about places where your feelings might have affected your response. Look for places that you left out part of the answer because a voice in your head told you that you were done and it was actually too early. You weren't done. You needed to give more information. Look for places where you might have had misgivings or felt something click, where you, instead of focusing on only what you did wrong, also try to really pinpoint things that worked, what you did right, what you want to repeat. And again, correlate your feelings with your score. Clarify the way feelings can be used or controlled. So, one of the biggest test strategies, not the study strategies, but one of the actual, you're taking the test, is controlling those feelings of anxiety or worry, right? You gotta just clear your head. A lot of times I tell people to take a deep breath, think about all the preparation you've done, and think very positively about your preparation and think positively about the test. And if you're still feeling really nervous, remind yourself that in the scope of life, this is just a test. It is not the end of the world. Your life does not depend on it. Nobody's life depends on this exam, right? You can always retake it. Worst case scenario, right? You fail and you retake the class. So, controlling that anxiety is one of the most important things you can do when you take a test. And of course, another strategy would be time management, right? If you don't know something, come back to it later. Don't spend all your time on one little thing. Focus your efforts on getting the majority of your credit. All right, now for chapter 11, the first part, which is what we're doing now, will be an introduction to hormones and their actions. So these are the physiology steps. Okay, then in part 2, we will go over a lot of names. I think it's about 28 hormone names. We'll say what organ produces them, which organ secretes, and what is the target organ, or even sometimes more specifically, which part of an organ is the target, and what effect does that have on the organ and or the entire body. Okay, so you definitely want to start memorizing. There's a lot to memorize. Okay. All right, a little review about hormones. They are produced by endocrine organs, but some endocrine organs are not purely endocrine organs. Some organs have mixed functions. For example, the heart. The heart's primary function is to pump blood throughout the body, so it's primarily an organ of the cardiovascular system, but it does produce a hormone called atrial natriuretic factor, and that's used to control blood volume, blood pressure. We will see this hormone again when we go over the cardiovascular system. Okay, so this is an example of how an organ can have a secondary function to produce one or more hormones. But you'll see other glands, like the pituitary gland, which its only purpose is to produce, to make hormones, make and secrete. Okay, so, sorry, I was chewing a Tic Tac so I wouldn't cough. Okay, so another thing to review is that hormones travel through the blood, which is coming in contact with essentially all of the cells in your body. But you don't want to send the same signal to every single cell, right? You don't want to turn every single cell on or off for a particular function. you're going to have a very specific target. So how this is accomplished is through the specificity of receptors, right? So you will have only a particular group of cells will have the particular receptor for a particular hormone, and so it's like a lock and key kind of deal. Okay, so even though that hormone is circulating throughout the whole body, and even though it's coming in contact with every type of cell, it will only actually interact and have its effect on the specific target. Again, due to the specificity of receptors to hormones. Okay, we call this slow because, of course, neurons are super, super fast, right? Milliseconds. Hormones will take at least an hour to circulate through the body, right? What else? Let's see. That's it for this. Okay. So now we're going to talk about the 4 categories of hormones, the 4 main groups. And the 4 main groups are going to be the amines, which are derived from tyrosine. And so here I have tyrosine. And remember, all amino acids have this structure at the bottom, right? You memorized that, chapter 2 perhaps. And we talked about how every amino acid will be identical on this region, and then its R group will be different, and that's what gives you the different amino acids. So for tyrosine, this is what it looks like. I'm not asking you to memorize this structure, just pointing it out. Okay, so we have different hormones that are derived from tyrosine. The adrenal medulla, remember the very middle of the adrenal gland, makes epinephrine and norepinephrine. And this is hydrophilic, meaning that it likes water, so it can dissolve in water, be carried through the blood. Okay. The thyroid gland, however, makes a hydrophobic hormone called thyroxine. Thyroxine has a very important role in regulating metabolism. We will talk more about its function in part 2 of this chapter. Okay, so it's interesting because these epinephrine, norepinephrine, and thyroxine, abbreviated T4, and also derivative or precursor rather, T3. They are derived from tyrosine, but they're made in different glands and they have different chemistry, right? So make sure that you're memorizing this correctly. All right. Also remember, the slang for epinephrine is adrenaline, but don't use that when you type your test answers. Make sure you use epinephrine, okay? And also, you can make a note here that in addition to tyrosine being a precursor for thyroxine, you also need iodine. The thyroid has to have iodine and tyrosine in order to make thyroxine, okay? And this is why iodine is added to table salt. If you go to the store and you just get a box of table salt, it'll usually say iodine added. Of course, nowadays we have such a big trend with sea salt, and most, if not all, of the sea salts will not have iodine. And people think this is a good thing, but iodine is really important for your thyroid to function, and we don't know for certain if we have enough iodine in our soil and therefore in our food. So goiters used to happen, just an enlargement of the thyroid because it's trying to produce something that it can't, right? And so then it's being told to do more, do its job more. but it can't do its job. And so then it just gets into this really huge, enlarged, overgrown state, and you can actually see a bulge on the side of a person's neck. And of course, this can lead to a lot of issues because now you can't regulate your metabolism the way that you should. So all sorts of problems can arise from that. In addition to, just a person's energy level and well-being. And so we started adding iodine, and it fixed this problem. And essentially no one in the United States has goiters anymore, but it's pretty common in other countries that don't have iodine in their salt. So I think it's probably still important. If you are big on sea salt, just make sure that you occasionally get a little table salt. If you have any processed foods, which I'm not encouraging you to eat processed foods, those are not good. But if you have a little bit, like a slice of bread, for example, it's probably going to have salt with iodine. So just keep an eye on it, okay? All right, moving on. Category number 2, polypeptide or small protein, okay? The polypeptide is a long chain of amino acids, right? And then a protein would be when that chain is folded. It's the most common category of hormones in the body, and they are also hydrophilic, meaning that they can interact with water, so they can easily be transported through the blood, right? And so one example that you're familiar with is growth hormone. Growth hormones produced from the anterior pituitary. We don't make a lot of growth hormone as adults, but we do produce some. And you might wonder then, how come we don't continue to grow? And the reason you don't continue to grow, even though you have low levels of growth hormone still, is because you won't make the receptors for the growth hormone. So even if you were to take a growth hormone supplement, it wouldn't have much, if any, effect because you don't have the receptors for it anymore, okay? But of course, when you're a child, right, and all the way up until you're an adult, then pretty much all of the tissues in your body are going to have the receptor and respond to growth hormone, okay? So, tissues in the body and bone even, right? Everything will respond to growth hormone until you're an adult. And another name for growth hormone is somatotropin. So you'll see that, I think, on one of the slides. So just take note. Somatotropin equals growth hormone, and then also know the abbreviation for growth hormone will be capital G, capital H. And in part 2 of the chapter, you're going to see a lot of names with their abbreviations, and for a couple, you will have a secondary name as well, okay? So a lot to memorize. All right, next category, glycoproteins. These are proteins that have carbohydrates on them, right? Glyco for carbohydrate. These are also hydrophilic. And an example from this category is LH and FSH, which I believe you're a little familiar with from anatomy. So remember that LH and FSH, first of all, LH stands for luteinizing hormone. FSH stands for follicle stimulating hormone. And these were first discovered and studied in women. And so that's why these hormones are named after the female reproductive system and their role in the reproductive system. However, men produce LH and FSH, and this is just as important for the male reproductive system as it is for the female reproductive system. We will learn all about that in the last unit, in fact, the last chapter of the course. Okay, and then finally, our last group of, or category of hormones are the steroids. And remember, we talked about a little bit about this already. Cholesterol is the precursor for all of your steroids. Your steroids include both sex hormones and non-sex hormones. Okay, so cholesterol is converted into different steroid hormones, and they are all hydrophobic. They cannot interact with water. Therefore, they cannot just go into the blood and travel through the blood. They have to be carried by albumin. And remember that albumin is a serum protein. It's the predominant serum protein in human blood plasma. And it's able to interact with water, and it's also able to interact with hydrophobic molecules. And so it's the perfect carrier. It carries things through our blood like calcium, sodium, potassium, fatty acids, hormones such as our steroid hormones, bilirubin, which you'll learn about when we go over the liver, as well as the thyroxine that we just talked about, T4, and some pharmaceuticals even are hydrophobic. And a classic example of that are barbiturates. And of course, the main function of albumin is to regulate this oncotic pressure, which is like osmotic pressure of the blood. And we'll talk about that later in the course as well. But you did see albumin when you looked at the cell transport, the cell membrane. When you looked at the PhysioX cell membrane experiment, and you looked at how quickly can things diffuse straight through the membrane, do they require a channel and therefore facilitated, or are they active? And you saw the albumin never crossed through the membrane because it was way too big. So even with active transport, you still cannot get albumin through. Albumin's too big, it will always stay in the blood. Okay, next, for steroids, it's really important that you realize that you cannot produce and store up steroids. They will immediately diffuse out of the cell, whatever cell is producing them. So the cell of the organ. And a lot of these are produced in the adrenal gland, okay, which you'll see in just a second. Okay, so steroids are all derivatives of cholesterol. They are all hydrophobic and require albumin to carry it through the blood, and they cannot be stored up. As soon as they are produced, they will diffuse out of the cell. Okay, so cholesterol is the precursor, and we can turn it into all sorts of different hormones. Okay, and the main hormones that you're concerned with are like progesterone, estradiol, which you know as estrogen, testosterone. Okay, and I believe this image here is only showing you the sex hormone pathway. It's not showing you cortisol. It's not showing you aldosterone. You do not need to memorize this pathway. This is simply to show you that cholesterol is always the precursor to every other type of steroid hormone. Okay, so now we're looking a little bit more specifically, and you'll need to start learning some of these specific pieces of information. I will never ask you the chemical structure. You just need to know cholesterol is the precursor. But now you're going to start putting together some hormones and some organs, okay? So the body will take cholesterol, and if you don't eat enough cholesterol, you can synthesize some cholesterol yourself. You shouldn't have such a fat-free diet that you don't have cholesterol. You should have some healthy fats in your diet, right? We talked about that before. You want to have your poly and your mono unsaturated fats, and you're going to want to have a little bit of cholesterol. Cholesterol typically comes from animal products, right? Eggs, dairy, meat. But you can also get some cholesterol from coconut. And if I remember, coconut's the only plant cholesterol, but I could be wrong. You guys could do a little research on are there any other plants that have cholesterol besides coconut? You can post that on your discussion. Okay, so progesterone is produced, if it's for men, it's produced in the testes, and even some in the adrenal glands. And for females, also some adrenal glands, and also the ovary, specifically the corpus luteum of the ovary. And it's okay if you don't know what that means right now. By the end of physiology, you'll know what that means. Okay, so men make progesterone in their testes and adrenal glands, and then that will get made into testosterone, which is also in the testes, specifically the outer, right, the Leydig cells kind of surround, they're in the interstitial cells of the testes, okay? And if you were a female, you would have testosterone that gets produced in the ovaries, and then the ovaries would be able to take some of that testosterone to make estrogen from. You do need testosterone, and you will use some testosterone just like men, but at much, much lower levels, right? And then you'll use most of your testosterone as a precursor to your estradiol or estrogen, okay? And then that'll be secreted by follicles of the ovaries. Now, what did I skip? Oh, cortisol. Okay, in the adrenal glands, for both men and women, the adrenals will produce your cortisol, okay? So cortisol is secreted by the adrenal cortex, right, the outer region. And cortisol is important to deal with stress. It can help control blood sugar levels, regulate your metabolism, help reduce inflammation, and even assist with some memory. Another hormone that's you need to know is aldosterone, which is also made in the adrenal cortex. You'll see aldosterone. If you don't see it later in this lecture, you will see it in part 2. Having a hard time remembering. But aldosterone has a role with salt and water balance, helps control blood pressure, and we will talk a lot about aldosterone when we get to a unit on the urinary system. Okay, so you do need to know progesterone. Where is it in men? Where is it in women? And same thing with cortisol, it's the same for men and women. Testosterone's different between men and women, right? And also want to point out that men will have small, low levels of estradiol and estrogen as well, but of course they don't have ovaries, so all of that has to take place in their gonads. Okay, that's good for now. Okay, so excuse me, continuing our discussion with steroid hormones, I talked to you about they're hydrophobic. You need a carrier protein to carry the hormone. And so you have a very unique pathway of action when it gets to the target, okay? And actually, it doesn't distinguish the target from the outside. It actually will go into any and every cell. And if that cell has the receptor inside the cytoplasm, then it will have its action. If it does not find a receptor, it will simply go back out and recombine with the carrier protein, and it will keep trying cell after cell until it's used up by target cells. Okay, so this is very unusual or interesting, if nothing else, that the receptors are hidden inside the cytoplasm of the cell. Okay, another thing that's interesting is that this receptor protein is linked to a special protein that will bind to DNA. So you get your hormone, right? It will just naturally pass through the membrane. If the receptor is present, it will combine with the receptor. Then you actually need 2 of these, and I'll show you on the next slide. Those will actually go into the nucleus and find their specific binding location. And that is how they will turn on transcription of this particular gene. And the transcription of that gene, of course, leads to translation, making your protein, and now you have the actual response. This hormone, whatever hormone this is, has told the cell to do some function by turning on one of its genes and causing it to transcribe and translate that gene into protein, and therefore you're adding function to the cell that it didn't have, or you're adding more of that function if it already had some going on. Okay, so it's pretty general, but there are still a lot of steps. Okay, and I mentioned that you need 2 of them, and we call that dimer. 2 of them is a dimer. And so we have the dimerization of the receptor. So you have 2 of these receptors, each with the steroid hormone bound to it, and those are linked to a binding domain that binds to the binding hormone response element on the gene. Okay, so this image is slightly confusing because at the top region is showing you the components, and then after the pink arrow, it's showing you the actual schematic. Okay, so at the top, it's just showing you all the individual pieces. You have a hormone, steroid hormone, it's hydrophobic, and then you have a receptor that we call the ligand binding domain, and the hormone is the ligand. It is bound or linked to a DNA binding domain, and you will need 2 sets of these. And now it's showing you the DNA here with a target gene, and on that target gene there are 2 hormone response elements, one for each receptor protein steroid, and when you get the dimer, which you see down here, each binding domain binds to each hormone response element. Okay, so if you only have one, nothing's going to happen, right? You need to have both hormones in their receptor so that each DNA binding domain can bind to the hormone response element on the target gene. And that is what will turn on transcription, and then of course all the steps after that to make the protein, whatever that gene was coding for. All right, you can copy paste this YouTube video to see this animation. Okay, So you need to know, how does the hormone increase transcription? It binds to a receptor with a DNA binding domain. The DNA binding domain binds to the hormone response element, but it first has to form you have to have a dimerization of the receptor, right? So 2 hormones, 2 receptors, 2 DNA binding domains, and each one binds to a hormone response element on the target gene. All right, next. Steroid hormone action. Okay, so everything I just talked about in both of those images, steroid will diffuse through the plasma membrane from wherever it was produced. It then binds to albumin or carrier protein, travels through the blood, and it will, just as it immediately diffused out of the cell that made it, it will diffuse into whatever cell it comes into contact with next. So it diffuses into a cell through the plasma membrane directly. And if the receptor is present, if this is the target cell, then the steroid will bind to the receptor protein in the cytoplasm and make the complex. And then you have 2 of these to make your dimer. Okay, so 2 complexes come together or dimerize, and then they will go into the nucleus and they will bind to the hormone response element on the target gene in the DNA. Then the target gene is activated, and that will cause transcription. Of course, then you leave the nucleus and then you have translation, and that's your action. Okay, so watch this video on your own. Okay, time for a little fun. I know that was heavy. Okay, this video is optional, but it's super cute. You should watch it. Laughing lowers levels of stress hormones and strengthens the immune system. 6-year-olds laugh on average 300 times a day. Kids, if you notice, they're always laughing. Adults, some don't laugh much, others laugh more, but not as much as children. Maybe we need to laugh, make an effort to laugh even more so that we can stay younger longer. Even just the anticipation of laughter causes a 27% boost in your mood. In the mood, elevating endorphins, right? Which may or may not actually change your mood, but I think most of you will agree when you know you're going to watch a funny movie or a comedy or something, you're going to play a funny game, you tend to just be in a better mood or have more energy, right? Smiling at each other feels good, right? It's the same thing, you're releasing endorphins. All right, so take time to smile. Okay, and laugh. Action of water-soluble hormones. So now we're switching gears from the steroid hydrophobic hormones. Now we're talking of hydrophilic. Okay, so polar hydrophilic hormones, they interact with water, but they cannot interact with the membrane. So they are stuck. They are stuck either inside or outside the cell. They cannot cross the membrane. So we need messenger systems. And recall you learned about second messenger system in chapter 7. It was actually, we borrowed Dr. Hayes' lecture, so you didn't hear it from me, but you still learned it, okay? And so you learned about one of the second messenger systems, the adenylate cyclase or cyclic AMP system. But today you will learn 2 more. You will learn phospholipase C calcium system, and you will learn the tyrosine kinase system. So again, remember that these are, all 3 of these are second messenger systems, but they get, I guess you could say they get more complex. Okay, so I'll briefly review with you, but you already know this. The adenylate cyclase CAMP system you will have your hormone from the outside bind to the outside of the cell at its specific lock and key fit, remember, specific receptor protein. And when that hormone binds to this protein, it causes a change in this protein to release these G proteins that are located at the bottom of the protein receptor. Okay, so that is kind of like pushing a button and causing a lever to fall. And that lever would be the G proteins, right? And so then those proteins activate the protein next to it, which is adenylate cyclase. Adenylate cyclase will form cyclic AMP by taking ATP, okay? And so, again, remember the whole point of this system is that the hormone cannot cross the membrane. So it binds to a receptor. The receptor activates G proteins, which are the second messenger system, and they will activate the adenylate cyclase. Okay, so the G protein activates adenylate cyclase, and then it has an enzymatic action down here where it uses ATP to produce cyclic AMP. And the cyclic AMP is the second messenger, okay? And its job is to go activate a protein kinase. And so it finds a protein kinase and it activates it. And then the protein kinase, now that it's active, is going to turn something on or off. It's going to either activate an enzyme to turn on some function, or it's going to inactivate an enzyme to turn off some function. Okay? And exactly which kinase it is and which enzyme it is, and if it's on or off, all of that will depend on the specific hormone and the specific receptor that it binds to. Okay? All right, so this is just a recap of everything I just said. So the hormone will find its receptor on the membrane, and the hormone is the first messenger, right? Sent through the whole body to find its target. The target, the receptor, will activate the G proteins, which will activate adenylate cyclase, which will turn ATP into cyclic AMP. And the cyclic AMP is the second messenger, which will activate a protein kinase. And the protein kinase will turn on or off some enzyme. Okay? Now for the new stuff. Phospholipase C, PLC, is an abbreviation that I think we made up here. I don't think that's in your book. But phospholipase C and calcium system. Okay, so the phospholipase C is going to lead to the release of calcium, which then will give the actual function or outcome. Okay, so this one's even longer. So get ready, take a deep breath. You can do it. Okay, so the hormone finds its specific receptor that will activate some G proteins. And this time, these G proteins are going to activate phospholipase C. Phospholipase. So this enzyme is going to, or this protein is going to have an enzymatic reaction to take a phospholipid from the membrane and split it. And it's going to split it into these two components called DAG and IP3. We don't care about DAG. I'm not going to ask you about DAG function. We don't know its function, okay? But what we care about is this IP3 subunit, okay? So the G proteins activate phospholipase C Phospholipase C takes one of the membrane, plasma membrane phospholipids and splits it. And then the IP3 is one of the components from that reaction, and it's going to go to the ER, the endoplasmic reticulum, and that's going to stimulate the release of calcium, a large amount of calcium released from the endoplasmic reticulum. And then unfortunately, this image doesn't show you the second part, but once calcium's released into the cytoplasm, it's going to activate this molecule called calmodulin. And calmodulin will then activate protein kinase. And of course, you know protein kinase will turn on or off some enzyme, some function, okay? So here's a recap of those steps. The hormone will find the receptor on the membrane. Remember, it cannot cross the hormone cannot cross the membrane because it is hydrophilic, okay? And it interacts with water but not fats, so cannot interact with the membrane. Okay, so it finds the receptor on the membrane. The receptor will signal the G protein, release the G protein to activate the phospholipase C. Phospholipase C will convert one of the phospholipids from the membrane into DAG and IP3. We don't care about DAG, we care about IP3. And then IP3 will cause the endoplasmic reticulum to release calcium, and this is calcium ions, Ca2+, Ca2+, okay? And then those calcium ions, Ca2+, will bind and activate calmodulin, and calmodulin will then activate protein kinase, and as you know, protein kinase will phosphorylate proteins like enzymes to give them a function, or turn it off, take away a function, okay? So that is the phospholipase C system. Now we're going to look at the tyrosine kinase action or system, okay? This is the last of the second messenger systems. Notice this interesting receptor. We have two parts. So this receptor in and of itself is a dimer. It's two parts put together, identical parts put together, okay? And in this example, we have the insulin receptor, and the hormone is insulin. So from the extracellular fluid, insulin comes in, and it binds to half of the receptor and the other half of the receptor. So it binds, so two half receptors form a dimer, and then insulin can bind to it, okay? So insulin is important for glucose and also amino acid transport into cells. So if you don't have insulin, it doesn't matter how much sugar you consume, it's not getting into the cell, and you're going to be very hungry. Okay, so note here, it takes one insulin molecule, but it takes two halves of the receptor to come together and dimerize. The receptor is also the enzyme tyrosine kinase. So that's why this system is called the tyrosine kinase action. And so the inside or bottom of this receptor is going to act as an enzyme. Okay. Active tyrosine kinase will phosphorylate a signaling molecule. Okay, so the first step was for the dimerization of the receptor and then insulin to come and bind to it. Second step and third step. Insulin, the binding of the insulin causes phosphorylation. We call it autophosphorylation because it's the same receptor. And it's going to take ATP to do this, okay? It's going to take ATP, and then it's going to phosphorylize some molecule, okay? So active tyrosine kinase of the receptor itself phosphorylates a signaling molecule. So you essentially have everything you need in the receptor. You just need to add ATP, and you need the signaling molecule itself. So the signaling molecule comes to the receptor, ATP comes to the receptor, and that's it. That's it. This actually seems a lot more simple than the last one. Step 4, now your signaling molecule is phosphorylated. It's going to start a cascade of effects. And with the example that we've been using with the insulin receptor and the hormone insulin, this cascade would end with glucose receptors being put into the membrane so that you can take up glucose. Okay, so let's review those steps. There are 2 subunits of the receptor that come together and form a dimer, okay? And the receptor is called tyrosine kinase. The receptor will autophosphorylate the tyrosine. Let me show you. So the tyrosine kinase is active with the hormone, and it's going to autophosphorylate the signaling molecule. Okay. So as soon as the hormone is bound to the receptor will be activated because it will autophosphorylate itself, and then it's going to phosphorylate a signaling molecule. So let's go over that one more time. Let's make that clear. So the first, step 2 and step 3, insulin binding causes autophosphorylation of the receptor. So these P's right here, autophosphorylized. So originally you did not have those phosphate groups there, but once insulin binds, you're going to phosphorylate the actual receptor, and then ATP is going to come by, and then you're going to phosphorylate some signaling molecule. So 2 phosphorylation steps, auto and then of the signal molecule. Okay. Now that you activate the signaling molecule, you're going to have a whole series of events take place that eventually lead to being able to uptake glucose. Okay, so now it's time for you to do some work. Hormone therapy consists of delivering medications, and this can sometimes be orally, and other times it needs to be through an injection, subcutaneous. So some hormones can be taken orally, and others must be delivered through injection. Tell me why. Now give me examples. Okay, so I trust you answered me. So you're correct if you are hydrophobic hormone, you cannot be ingested, right? So hydrophilic, sorry, let's start with hydrophilic. I know I didn't type that on the slide for you, but hydrophilic polar molecules cannot cross the membrane. These are the ones produced by the adrenal hormones, and so you would not be able to take these orally. So can't take a pill for hydrophilic molecules because it will just be digested. Okay, but when it's fat soluble or hydrophobic, those are the ones you can go ahead and just take as a pill. And that makes sense because steroids are hydrophobic. Steroids include sex hormones. Birth control pills are have sex hormones, and you can take birth control pills, right? Orally. So that will always help you remember. Okay. Another thing, if you remember that epinephrine is hydrophilic, and when people have a severe allergy, they carry around an EpiPen, and the EpiPen is a shot, right? You stab, not really stab, it sounds dramatic, but you, well, I don't know any other synonym right now. So you're going to stab that EpiPen somewhere in like their thigh, right? And it's going to deliver epinephrine to them very quickly. And this is, again, you cannot take the epinephrine as a pill. It will not work. Insulin also, right? Diabetics, there's no such thing as an insulin pill. It has to be administered through a shot. This is not because they need it super quickly. It's because it will not work if you take it as a pill. Now, we do have infertility shots, which is confusing because those are sex hormones and they can also be taken through pill. But this, I think, has to do with the dosage, the timing with a female cycle, and yeah, just getting you here. It is more of a timing issue and a dosage issue. But when it comes to like taking your birth control pill, you can take a pill every day, or you can wear a patch, right? There's different delivery methods. It's not crucial that it's taken one way or the other when it comes to the steroid hormones. And then thyroid, same thing. You can have it as a pill because it's also hydrophobic. So the hydrophobic, like your steroids, your thyroid pills, birth control pills, right? The tricky one here is the infertility shots, okay? So please make a note of that. All the other shots, they are shots because they have to be. You would not ever be able to have that as a pill. Okay, so hydrophobic is fat soluble. It's able to cross the membrane directly. It includes your steroids, your thyroid hormone, and you can take those as a pill. Okay, your adrenal hormones, which are hydrophilic, you cannot take them as a pill. They'll be digested. Okay. All right, hormone interactions. We are almost done. Synergism means that it's better together than apart. You cannot just double the dosage of one hormone and get the same effect. You need the two together, okay? An example of this is testosterone and FSH, which will make more sense when we study the reproductive system. Okay, the permissive effect. One hormone will actually enhance the effect of another hormone. Thyroid hormone and epinephrine are examples of that. And then, of course, there can be antagonistic interactions where one hormone will decrease the ability of another hormone to bind to its receptor. And glucagon and insulin have this antagonistic effect on each other. Okay, and that makes sense because they have opposite signals, right? Insulin is telling your body to put all the glucose transporters into the membrane and take up all the glucose. But glucagon is telling the body your glucose is all gone, right? And you need to start breaking down your reserves and releasing it. All right. Last slide. Receptor regulation. You can decrease or down regulate the amount of receptors that are in a membrane, and that will, of course, if you reduce receptors, then you're going to reduce the effect that a hormone can have on its target. So the concentration of receptors for a hormone decreases, and then even though you have the hormone present, it doesn't have anything to bind to and it won't have any effect. Unfortunately, we see this with type 2 diabetes, where you produce plenty of insulin, but you no longer produce enough receptors, and so it cannot do its job. And we believe that this has to do with excessive amount of adipose tissue, and that's something you could also look into a little bit more for your discussion. We will talk about diabetes later in more detail in this class, but that would be an interesting topic. Okay, upregulation, of course, can happen as well. You can increase the amount of receptors, and therefore even just a little bit of the hormone will have a greater effect because you will bind it more readily. So you increase the sensitivity to a hormone. We see this with our gonadotropin releasing hormone, GnRH, increases sensitivity of the anterior pituitary to more of that hormone. So we could call this a priming effect, getting it ready to be more sensitive. And it doesn't always have to be to the same hormone. It could be priming it for a different hormone, right? So, yeah, that's it. Get ready for part 2. Lots of hormone names. All right, bye.